Many people believe that it isn't likely that a blood transfusion will affect them in any significant way. Well, unfortunately, I have some bad news. In an October 2nd article of the McClatchy Tribune Business News, it was reported that Dr. Bruce Lennis, the medical director of the community blood centers, said there is an 85% chance you or someone in your family will need a blood transfusion. That's an 85% chance of a blood transfusion directly affecting you. So I'm sure you'd like to be confident in the fact that if and when you ever need a blood transfusion, the blood will be readily available. Unfortunately, that may not be the case. The amount of blood being donated is struggling to keep up with a medical demand, and that is resulting in a blood shortage. As a mechanical engineer who plans to focus in bioengineering and eventually study bioengineering in graduate school, this is a problem that concerns me a great deal and led me to research possible solutions. Today, I would like to talk about the blood shortage, a possible scientific solution, arguments against this possible solution, and evidence to show that the solution I suggest will indeed be a viable option in the future. So to start with, I would like to talk about the blood shortage itself. Currently, blood banks have a low supply of blood, and this problem is likely only going to get worse. As you can see from this graph that I've created from data from the America's Blood Centers, North America's largest network of community blood banks, that 30 to 45% of the centers had a low supply of blood between the months of July and October of this year, meaning they had a 2-day supply or less. This is a very dangerous situation, and if there was ever to be any sort of large-scale disaster, these banks would have a difficult time supplying enough blood to all of those people that need transfusions. So, what is the cause of this shortage of blood? Well, the answer is a lack of blood donors. According to the AABB, a standards organization that works closely with the US FDA and was formerly known as the American Association of Blood Banks, only 39% of the population is actually eligible to donate blood, and less than 10% do. 15 million units of blood are donated each year, and over 14 million units of blood are used in transfusions each year. This is a very small margin of error. And also, blood has a short shelf life. Red blood cells can only be stored for 42 days, so we need a constant supply of blood. Unfortunately, we don't usually receive blood in a constant amount because during the winter and summer months, colleges are not in session, and college students are much less likely to donate blood while they're on their breaks. Also, according to the AABB, people aged 69 and over make up only 10% of the population, but they account for 50% of all blood transfusions. As baby boomers start to reach this elevated age, we're going to have a larger percent of the population in that age group, and therefore we're going to have an increased number of transfusions. So clearly this is a huge problem and something needs to be done. But what can we do about it? Well, the short-term solution would be to try and increase blood donors. But the recent low blood donor turnout rates show that we're likely going to need a long-term scientific solution for this problem as well. And in my research, I discovered that stem cells provide the opportunity for a long-term solution to the blood shortage. Now, the most commonly heard about types of stem cells are embryonic stem cells. And according to the National Institutes of Health's stem cell information page, these embryonic stem cells had the amazing ability of being able to transform into any other type of body cell. You can see the amazing potential that this has because we could just transform our stem cells into blood cells for use in transfusions. So, what scientists will do is take stem cells from young embryos called blastocysts. As you can see in this image here at the top, a blastocyst is a mostly hollow body of cells, and attached on one end is a small group of cells called the inner cell mass. And this inner cell mass is made up of the stem cells. Now, when allowed to develop normally, these stem cells will fold up and form 3 germ layers: the ectoderm, the mesoderm, and the endoderm. And from these 3 layers, all the cells, tissues, and organs in the body will form. What scientists do, however, is remove these stem cells from the young embryo, and they can place them in a solution called a culture medium. And in this culture medium, the cells will divide and multiply without differentiating. So just to illustrate this, in this slide here, you can see the stem cells being removed from the young embryo. This embryo is actually an 8-cell embryo, not a blastocyst. It's a younger embryo, but they're still stem cells. And they will take these cells and place them in a culture medium where they will multiply and divide and form colonies. And the colony you can see here is in the center of the image. What scientists will do then is add different genetic genes and change the chemical composition of the culture medium, which will cause the cells to start to differentiate. For example, here we see red blood cells that were created from stem cells. And what scientists can do is experiment and learn what changes created what different types of cells and record these changes so they can later create the types of cells they want. So now that we have talked a little bit about how the scientists can create red blood cells from these cells, I'd like to talk a little bit about the ethical debate regarding this, because the problem with this method is that it results in the destruction of embryos, which many people are opposed to, understandably so. Fortunately, there is good news. In an article from McClatchy Tribune Business News titled "A Breakthrough, Then a Surge in Stem Cell Research," it was reported that scientists have learned how to create cells that are almost identical to embryonic stem cells without actually destroying the embryos. And they do this using skin cells, believe it or not. And the scientists can take adult skin cells and genetically reprogram them to create cells called induced pluripotent cells. And these cells have the same ability as embryonic stem cells to turn into almost any other type of body cell. Now, this is a huge breakthrough, and as this technology is developed and enhanced, we can hopefully eliminate the need for embryonic stem cells and effectively end the controversy. So now that I've touched on the cultural debate and the ethical debate regarding this stem cell research, I'd like to talk more about how we can use stem cells to end the blood shortage. In the last year, scientists have been able to improve their efficiency at creating red blood cells from stem cells. And this was reported in an August 20th article of the Los Angeles Times. They reported that a biotechnology company called Advanced Cell Technology was able to create 100 billion red blood cells from a single plate of embryonic stem cells. That's enough red blood cells to fill 2 to 3 test tubes full of blood. And while this method is 100 times more efficient than any other effort to create red blood cells from embryonic stem cells or any other type of stem cells. And according to the United Kingdom's The Times newspaper, in an article also from August 20th, these cells had the ability to transport oxygen, and 2/3 of them had shed their nucleus, which is a sign of being fully mature adult red blood cells. And the scientists hope that they can raise this number closer to 100%. According to Robert Lanza, the lead researcher of this project, there is still a long way to go, but we could actually see clinical testing of these red blood cells in blood transfusions within the next year or two. So they're making huge advancements in this technology, and I'm looking forward to see what comes up in the upcoming years. So just to recap briefly, I've talked about the blood shortage problem. I've talked about the possible solution that stem cells provide. I have talked about the ethical debate regarding stem cells and the new technology that will hopefully eliminate that. And I've also discussed the recent advancements made by scientists to create blood that can possibly be used in blood transfusions. Stem cell research is still in its infancy. We likely have a few decades yet before we'll be able to convert stem cells into blood cells efficiently enough to be commonly used in transfusions. However, the strides they are making are enormous, and while the blood shortage is still a huge challenge, stem cell research can help ensure that it is not an insurmountable one. Thank you.